Good morning, friends. Today we are going to explain excitation, contraction, coupling. This is important topic for the B.Sc. third year in paper first animal physiology. Then this topic has been explained in front of you by myself, Prem Singh Chauhan, Associate Professor in Zoology. As we know, the skeletal muscle fiber is so large that action potential is spreading along its surface membrane cause almost no current flow deep within the fiber. Maximum muscle contraction, however, requires the current to penetrate deeply into the muscle fiber to the vicinity of the separate myofibrils. This penetration is achieved by transmission of action potential along transverse tubules, T-tubules that penetrates all the way through the muscle fiber from one side of the fiber to the other. This has also been illustrated in this diagram. The T-tubule system action potential cause release of calcium ions inside the muscle fibers in the immediate vicinity of the myofibrils, and then these calcium ions then cause contraction. This overall process is called excitation contraction coupling. The transverse tubule, that sarcoplasmic reticulum and myofibrils are shown by this diagram. This system this is sarcolemma and terminal cisternes of the sarcoplasmic reticulum system. Transverse tubules, mitochondria, sarcoplasmic reticulum, and again transverse tubules. Along with these are the myofibrils. This is triad of the reticulum in adjacent to the myofibril. The myofibril is continuously formed by A band and I band. A band consists of H zone in middle, which is again bifurcated by the M line, and I band is divided by Z line in between, and there is a sarcotubules. This is all subcomponent of this sarcoplasmic reticulum or transverse tubule system. The T-tubule sarcoplasmic reticular system. The T-tubules are small that runs transverse to the myofibril. This is transverse tubules. They begins at the cell membrane and penetrate all the way from one side of the muscle fiber to the opposite side, where the T-tubules originates from the cell membrane and they are open to the exterior of the muscle fiber. Therefore, they communicate with the extracellular fluid surrounding the muscle fiber and contain extracellular fluid in their lumen. In other words, the T tubules are actually internal extensions of the cell membrane. Therefore, when an action potential spreads over a muscle fiber membrane, a potential change also spread along the T tubules to the deep interior of the muscle fiber. The electrical current surrounding these T tubules then elicits the muscle contraction. This diagram also shows a sarcoplasmic reticulum in yellow. This sarcoplasmic reticulum is composed of two major parts. First, large number of terminal cisternae. These are terminal cisternae represented by the yellow colors that about the T tubules and along longitudinal tubules that surrounds all surface of the actual contracting microfibrils. Release of calcium ions by the sarcoplasmic reticulum. Release of calcium ions by sarcoplasmic reticulum. Once the special feature of the sarcoplasmic reticulum is that within its vesicular tubules is an excess of calcium ions in high concentration, and many of these ions are released from each vesicle when an action potential occurs in the adjacent T tubules. This diagram shows sarcoplasmic reticulum with calcium ions. These calcium ions are released. These are DHP receptors, and this is action potential. This shows the action potential of the T tubules cause current flow into the sarcoplasmic reticulum cisternae where they about T tubules. As the action potential reaches the T tubules, the voltage channel is sensed by dihydropyridine receptors, DHP receptors, dihydropyridine receptors that are linked to the calcium release channels and also called ryanodine receptor channels in the adjacent sarcoplasmic reticular system. Activation of dihydropyridine receptor triggers the opening of the calcium release channels in the cisternae as well as in their attached longitudinal tubules. These channels remains open for a few milliseconds and releasing calcium ion into the sarcoplasm surrounding the myofibrils and causing contraction. The calcium pump removes calcium ion from the myofibrillary fluid after contraction occur. Once these calcium ions are released into the lumen of the sarcoplasm, there is a generation of contraction. Once the contraction has occurs, the calcium pumps again removes calcium ions from the myofibrillary fluid after contraction occur. Once the calcium ion has been released from the sarcoplasmic tubule and have diffused among the myofibril, muscle contraction continues as long as the calcium concentration remains high. However, continually active calcium pump located in the vas of the sarcoplasmic reticulum pump calcium ions away from the myofibrils back into the sarcoplasmic reticulum. This pump can concentrate the calcium ions about 10,000 folds inside the tubules. In addition, inside the reticulum is a protein called calsequestrin. This is the protein calsequestrin that binds up to the 40 times more calciums. Excitatory pulse calcium ions. The normal resting state concentration is less than 10 to the power minus 7 moles per liter of the calcium ions in the cytosol. That baths the myofibril is too little to elect contraction. Therefore, the troponin tropomyosin complex keeps the actin filament inhibited and maintain a relaxed state of the muscle. Conversely, full excitation of the T tubules and sarcoplasmic reticular system causes enough release of the calcium ions to increase the concentration in the myofibril fluid to as high as 2 is multiplied by 10 to the power minus 4 moles per liter, a 500-fold increase, which is about 10 times the level required to cause maximum muscle contraction. Immediately thereafter, the calcium pump depletes the calcium ions again. The total duration of this calcium pulse in the usual skeletal muscle fiber lasts about 1/12 of a second, although it may last several times as long in some fibers and several times less in others. In heart muscles, the calcium pulse lasts about 1/3 of a second because the long duration of the cardiac action potential. During this calcium pulse, muscle contraction occurs. If the contraction is to continue without interruption for the long intervals, a series of calcium pulses must be initiated by a continuous series of repetitive action potential. Excitation contraction coupling showing action potential that cause the release of calcium ions from the sarcoplasmic reticulum, then uptake of the calcium ions by the calcium pump. This is sarcolemma. This is action potential, and this one is calcium pump. ATP require these calcium ions, and this one is actin filament and myosin filaments. This thin one are the actin filament and thick one is the myosin filament. Excitation contraction complete in a muscle showing an action potential that causes release of calcium ion from the sarcoplasmic reticulum. The uptake of calcium ions by ATP adenosine triphosphate. The summary of entire process has been shown by this diagram. This is quartal release of acetylcholine, generation of the end plate potential, transmission of the action potential on the sarcolemma, the movement of action potential in the T-tubule system, then voltage channel sensed by dihydropyridine receptors, then release of calcium via ryanodine receptors, which initiates the contraction of myofibrils. These all process is releasing calcium ions and initiation of the contraction. The molecular events of the contractions are the calcium tends to bind to the troponin C. As we know, troponin is the protein which is having three units, TPT, TPI and TPC. TPC units is calcium binding units, so the calcium is binds with the TPC. Then conformation change in the tropomyosin and exposure of the binding sites on the actin. Then myosin head binds to the active site, and in the last one is the power stroke or muscle shortening. This all occurs during the muscle contraction. This diagram summarized two systems at the same time. One's molecular events of the muscle contraction and release of calcium from the sarcoplasmic tubular system. This is all about the excitation contraction coupling. Thank you for watching this video. Have a good day. Please do like, share and subscribe